Excitation, contraction, coupling. This process is the stimulation of muscle contraction of an individual muscle fiber, which occurs at the sarcoplasmic reticulum junction. The sarcolemma is the cell membrane, and the sarcoplasmic reticulum is a system of membrane-bound tubules that surrounds muscle fibrils. The red area shown is the sarcolemma. The tan area is the motor end plate at the end of a nerve. The reaction starts at the axon terminal of a motor neuron. A nerve impulse goes down the axon terminal into the motor end plate, where a nerve and a muscle meet. This nerve impulse causes voltage-gated calcium canals to open. When these canals open, calcium ions are released. The releasing of calcium ions causes synaptic vesicles in the motor end plate to release acetylcholine into the synaptic cleft. This chemical causes other chemical reactions to occur. After the acetylcholine crosses the synaptic cleft, it reaches ligand-gated ion channels and nicotinic adesyl receptors, also called sodium and potassium ion channels. These two types of receptors use acetylcholine to open. Once these channels open, small amounts of potassium is released and large amounts of sodium is absorbed. The potassium is symbolized by the blue pentagons, and the sodium is symbolized by the purple triangles. This influx of sodium and potassium causes a depolarization of skeletal muscle fibers called end plate potential. The RMP changes from -90 MV to -70 MV. If the opening of these canals creates enough end plate potential, then it will start opening voltage-gated canals. This results in muscle action potential, which is the electrical activity inside a muscle cell during physical activity. Muscle action potential moves across the end plate, across the sarcolemma, and down the transverse tubules, towards the next step of excitation-contraction coupling. The action potential that moves down the T-tubules causes the voltage-gated calcium ion channels in the sarcoplasmic reticulum to open and release calcium. These channels are also called ryodyne receptors and hydropyridine receptors. The calcium then goes down into the sarcomere and connects to troponin. A sarcomere is a functional unit of contraction within a muscle fiber that consists of actin and myosin filaments. This calcium is then used for the contraction cycle. This cycle is when the muscle fiber generates tension through the action of the actin and myosin crossbridge. There are several items involved with this process. Actin, a thin filament protein, myosin, a thick filament protein, troponin, a protein molecule, tropomyosin, a two-stranded coiled protein, a myosin head, part of the myosin which attaches to actin, a crossbridge, an area where the myosin head sticks out and attaches to actin, adenine triphosphate, which is a source of energy, adenine diphosphate and phosphate, the broken down version of adenine triphosphate. Lastly, there is an active site on actin. This green circle shows the attachment places for the myosin head on the actin strand. There are 4 steps to this process. The first step involves the myosin head uncocking itself and kicking off the extra phosphate. In the second step, the myosin head then attaches itself to the active site on the actin and removes the extra ADP. Then it power strokes. Power stroking is the movement of the myosin head along the actin filament. The third step is when the myosin head adds ATP so it can detach itself from the active site on the actin. In the final step, the myosin head recocks itself by breaking down ATP into ADP and phosphate. After excitation-contraction coupling is complete, the muscle relaxes and all chemicals are reabsorbed.